Hi everyone, thanks for checking out this video. So in the last lesson, we read our gene expression dataset into Python, and we did a little bit of like processing and filtering, leaving us with normalized expression data for 11,000 or so genes. And in this lesson, we're going to finally do the analysis that we've been building up to in basically this whole course so far. We've been building up to this differential expression analysis that I've been telling you guys about. And in this video, we're going to finally actually do this analysis. So just to remind you guys what the point of all of this is. It's like I was showing you before on the slides. The point is that we have, for every gene, basically, an observed distribution of expression measurements in both the IPF and control conditions. And the goal of a differential expression analysis is to look for genes where there's an interesting difference between the observed expression distributions in the two conditions, basically. And actually, at this point, this slide looks a little bit unrealistic because remember, at this point, we've actually normalized the data with this CPM normalization correcting for sequencing depth. So at this point, it's not these like integer counts anymore. We actually have counts per million, which is a type of normalization. But the idea is still the same. We're going to try to use a statistical test to look for genes where there's a difference between the two observed distributions in the two different conditions, basically. So to do this, we're going to use a statistical test called the Kolmogorov-Smirnov test, or the KS test for short. So yeah, the KS test is basically a test of whether two observed distributions were drawn from the same underlying distribution, or if they came from different underlying distributions. So basically, in the KS test, the null hypothesis is that two observed distributions, in this case, for us, it'll be the IPF distribution for a given gene and the control distribution for a given gene. The null hypothesis is that these two observed distributions came from the same underlying distribution. And the alternative hypothesis is that they came from two different underlying distributions. So basically, it's like we talked about in the statistics video. The goal is going to be for every gene to generate a p-value telling us the probability of seeing as much of a difference between the observed distributions as we see, given the null hypothesis that they came from the same underlying distribution. We want to generate a p-value for every gene, testing that, basically. I'll tell you guys the same disclaimer that I told you in the last video, too. The point of this whole course really is to be educational, and that involves making some simplifications so that I can like teach these concepts to you guys. And yeah, the use of the KS test is another simplification, because I mentioned here too that in real life, this type of analysis would not actually be done with the KS test. So it's a little bit unrealistic to use one here because in real life, it's not actually the smartest way to do this. So yeah, I didn't actually pick the KS test because it's like the best way or the smartest way. I picked it because I thought it would be like the simplest way and the easiest way to teach this concept to you guys. But in real life, yeah, there's these R packages like DE-seq2, EdgeR, and limavoom, and these packages use sort of like more advanced statistical methods that have been thought up by people way smarter than me. So yeah, they actually have like more like thought out and optimal ways of doing this statistical test, basically. So yeah, the KS test is not really realistic. It's actually like a bit too underpowered to be used in real life, meaning it's not good enough at like detecting real differences. But yeah, I didn't pick it because it's actually like the best. I picked it because it's the simplest and the easiest for teaching this topic. So it might actually be yeah, like a bit unrealistic compared to what's used in real life. But yeah, for teaching purposes, for like educational purposes, I think it'll be okay for this video. Another thing to stress is that the KS test is not a test of a difference between means. It's a test of a difference between distributions. So yeah, I just did a Google image search of like bell curves with different standard deviations. So for example, all of these different distributions in this image have the same mean but different standard deviations, and the KS test would identify these as being different distributions if you had enough samples drawn from each one. It would give you a low p-value and suggest that these are different distributions, even though they have the same mean. So yeah, this is not a test of a difference between means. So it is possible to have two distributions with the same mean still being identified by the KS test as different distributions. So we'll account for that too in a couple minutes. So in a couple minutes, we're actually going to use a different measure to compare the means of the two conditions, the IPF condition and the control condition. So first we'll get the p-value for each gene, and then I'll show you guys a different way to get a comparison of the means for each gene, basically. Okay. So let's give this a try. So I think like usually when I'm doing something like this, where like eventually we're going to be writing like a for loop to go through and calculate these things for every gene. But before we do that, we're going to make sure that we can correctly get these measurements for just the first gene. So yeah, this block of code here is basically just taking the first gene, which is row 0 in our data matrix. So yeah, pulling out the measurements for just the first gene, first for the control samples, and then next for the IPF samples. So yeah, basically just pulling the control and IPF measurements out just for the first gene, so we can try to get a p-value for them, basically. And then, yeah, like usual, just a little sanity check, making sure that these have the correct shape. Yeah, so 103 samples each. And then we can do the KS test with this SciPy stats function. And yeah, this will do the KS test and then give us the p-value. And yeah, so it happens to be the case that for the very first gene, it looks like the two observed distributions for the control and IPF conditions actually were pretty different, because this p-value is very low here. Again, it's scientific notation, so the p-value isn't 3.2 something something. It's like 0. A bunch of zeros and then 3, 2, 6, 4 something something. So yeah, don't forget the scientific notation here, basically. But yes, that's our p-value. Easy enough. And then, like I told you guys, the other thing we need to get is a comparison of the means for the two conditions for this first gene. Because yeah, a low p-value for the KS test does not automatically mean that the means are different. But yeah, in terms of looking for like biologically relevant differences, it's not going to be enough just to have a low p-value on the KS test. We're going to look for genes that have both a low p-value as well as a difference in means that will maybe hopefully suggest something about like biological relevance. And the way we're going to put a number on that is with something called the log2 fold change. So yeah, this is something you see in papers a lot. Yeah, like papers doing like gene expression analysis. Yeah, they often use something called the log2 fold change to talk about the difference in means between different conditions in terms of gene expression. So yeah, it looks like a little bit of confusing math, but it's actually not that different. So yeah, you know what? Maybe I'll just type some things out to kind of explain what's going on here. So the idea here is first of all, we're just gonna take the ratio of the mean of the IPF expression for this gene over the mean control expression for this gene. So this by itself would just be called the fold change. The fold change is just the mean of the IPF samples divided by the mean of the control samples for this gene. So yeah, that would just be like a straightforward comparison of how high or low was the gene expression in the IPF samples compared to the control samples in terms of their means, basically. And then there's this log2, which seems like maybe like some confusing math at first. So just to remind you guys what log2 means. So to take the log2, and I'm just going to write it like this because I can't easily do the underscore typing, but just to take the log2 of some number x means basically what number do we need to exponentiate 2 to so that it will equal x. So if x was if x was 4, then we'd say, okay, what number do we need to put 2 to in order to get 4? And the answer is 2, because 2 to the second power would give us 4. And if we take the log2 of 8, then we'd say, okay, what number do we need to put 2 to get 8? And then the answer would be 3, because 2 to the third power equals 8. And then if we took the log2 of 1/2, that would be -1, right? Because 2 to the -1, remember when you have a negative exponent, you just take the number and like put it on the bottom of a fraction. So it would be like 1/2, which would be 1/2. So yeah, the log2 of 1/2 would be -1. Okay, so why are we doing this though? Why are we dealing with this log2? Isn't that just making it like more confusing? Well, I think like maybe the easiest way to explain why we're going to use this is with an example. So let's say we have a gene where the mean of the IPF condition is 4, and then the mean of the control condition is 2. Yeah, for our example gene, yeah, the mean of the IPF samples for this example gene is 4, and then the mean of the control samples is 2. So that means the fold change, the mean of the IPF over the mean of the control, the fold change is 4/2, which is just 2, right? We'll call that example gene 1. But then let's say we have another example gene 2, where the mean of the IPF samples is 2, and the mean of the control samples is 4. So for example gene 2, the fold change is 2/4, which is 1/2. So it seems like both of these example genes have a change in mean expression between the conditions that basically has the same effect size, right? Because in both of these example genes, one condition has twice the mean expression of the other condition. So example gene 1, the IPF condition has twice the mean of the control condition. And in example gene 2, the control condition has twice the mean of the IPF condition. So it seems like these effect sizes are basically the same effect size, but just in opposite directions, right? Yeah, it seems like a similar size of the effect, just going in opposite directions, where in both cases, one condition has twice the mean of the other condition. So ideally, we'd want a measure of the effect sizes where both of these example genes would have like the same effect size, just in opposite directions. But the fold change by itself doesn't accomplish that, because 2 and 1/2 aren't like symmetrical in a way that they're the same effect size, just in different directions. They're not really comparable in like a symmetrical way like that. But we can use log2 to actually solve this problem for us. So what happens when we take the log2 of the fold change for this first example gene? So the log2 of the fold change 2 equals 1, right? Because what number do we need to raise 2 to in order to get 2? The answer is 1. So the log2 of 2 is 1. And then for the second example gene, if we take the log2 of the fold change, the log2 of 1/2, yeah, even that was one of the examples up here. The log2 of 1/2 is -1. So do you guys see this cool usage of log2 here? How it basically turns the fold change into a measure of effect size that is symmetrical around 0. So basically, if one of the means is twice the other, that will be either 1 or -1, depending on which direction the change was in. And if one of the means was 4 times the other, then the log2 fold change would be either 2 or -2. And if one of the means was 8 times the other in either direction, the log2 fold change would be 3 or -3, depending on which direction the change was in, whether the control or the IPF was 8 times the other. So yeah, the reason we're doing this like seemingly confusing math here with log2 is basically to turn this fold change comparison of means into a effect size measurement that is symmetrical around 0, so that the absolute value of a change in either direction will be the same, but it'll just be like positive or negative depending on which one was like twice the other, for example. Okay, so this is a pretty good measure of effect size, but there is one problem. So I wonder if you guys can maybe like pause for a second and maybe without reading ahead, see if you can figure out what the problem is here. If you want to like quickly pause and think about it. Okay, so the problem is this will give us an error if either one of these means is 0, because we can't divide by 0 and we also can't take the log of 0. So if either condition has a mean of 0, this will give us an error when we try to calculate it. And the way we're going to deal with this is a little bit of a trick. It's a little bit of a hack, to be honest. And yeah, if any of you guys are pure mathematicians, then I apologize for what you're about to see, because you're probably not going to like it. And to be honest, I also didn't like this at first. To me, it seemed like fishy and like not rigorous, or something, or just like not. It didn't seem like a good thing to do to me. But this is the way that this issue is typically solved in real life. I didn't like make up this trick. This is what like the actual packages like actually do. So yeah, I didn't make up this trick. This is like a somewhat widely used trick to get around this like divide by 0 and log of 0 issue. So the trick is that we're just going to solve this issue by adding a small number to both the top and bottom of this fraction, basically. So both the numerator and the denominator will add some small number called epsilon, which could be like 1 or 0.1 or 0.01. And adding a small number to both the top and bottom of this fraction will solve this like potential divide by 0 or log of 0 error that we might get. Well, yeah, we'll call it epsilon. I'll make it 1. You guys could make it like 0.1 or 0.01, or really whatever you want it to be. And then we'll calculate the log2 fold change like this, basically. We're basically just take the mean of the control samples and the mean of the IPF samples. And then the fold change will be basically this fraction, except adding epsilon to both the top and bottom of the fraction. And then once we have the fold change, we'll take the log2 of that, and that will be our log2 fold change. Also, yeah, here it is. Here it is for the first gene that we were working with. Okay, so a quick note about this. Yeah, at first I didn't like it. I thought this was fishy and like not a good thing to do because we're changing the numbers around. We're making this fold change something other than what it actually is by adding a constant to both top and bottom of the fraction. So I thought that was fishy at first, and I didn't like it. It seemed to me like not mathematically rigorous. But after thinking about it a bit, I kind of changed my mind, and I became more okay with doing this, and more okay with like the fact that this is kind of how it's done in real life. And what made me change my mind is I just thought, like, what is the goal of this analysis? What are we trying to do here? Are we trying to write a mathematical proof? Are we trying to do like an airtight like mathematical derivation? No, we're not. We're not trying to write some like totally rigorous, solid mathematical proof here. What we're trying to do is look for genes that are interesting. The goal here is not to write a mathematical proof, it's to find genes that have an interesting difference between these conditions. And so when I thought about that, I thought, okay, so given that that's the goal, I do think this little like hack is actually okay, just to get our code to run without an error so that we can identify genes that have a interesting difference between the conditions. The other thing too is that I originally didn't like that this epsilon trick basically changes the numbers. So it changes the fold change to be something other than what it actually is. But then I realized that it actually changes things in a way to make this measurement even more conservative and even less likely to like give us false positives and like falsely identify a gene as being different when it's not actually different. It actually makes that less likely to happen, because if you think about it, what happens when we add a number to both the top and bottom of a fraction? We basically dilute that fold change by a little bit. So first of all, let's just think, what would this log2 fold change be if both conditions had the same mean? Then we'd be taking the log2 of 1, which would be 0, right? So if both the top and bottom of the fraction were both the same, they both had the same mean, then this log2 fold change would be 0. So what would happen if we add a very, very large number like 1,000,000 to both the top and bottom of this fraction? Let's say that it was a mean of 4 for IPF and a mean of 2 for the control. And then what would happen if we added a very large constant to both the top and bottom of the fraction? Then we'd be getting a fold change close to 1, right? Because the top and bottom would be almost the same, and then we'd be getting a log2 fold change of close to 0. So actually, yeah, that's like an extreme example, but even adding like a small constant, the effect it would have, if any, would be to make the difference between conditions slightly more diluted. So it wouldn't be likely to give us more false positives or like more genes being detected as having a difference when they actually don't. If anything, it makes the analysis more conservative by very slightly diluting the difference between conditions so that more of a difference would be required in order to pass the threshold we're going to eventually use. So yeah, after thinking about that, I became more okay with this epsilon trick. Again, yeah, I didn't make this up. This is how it's actually done in some of the like real life packages even. But okay, so yeah, at this point we've gotten for the first gene the p-value for the KS test, and we've also gotten the log2 fold change, which is the effect size for the difference between means, basically. So yeah, now that we've gotten that for the first gene, basically what we want to do is just write a big for loop to go through and get these two things for every gene that we're going to be testing. So yeah, this is basically just like taking what we've already done and just putting it inside a big for loop to go through and do these calculations we just did for every gene that we're going to be testing. So yeah, just going to be defining some empty lists here to save our p-values and log2 fold changes. Again, epsilon is like small constant to avoid division by 0. We're taking the log of 0. And then just a for loop to go through every row in our data matrix. And then for that row, we'll split up the control and IPF conditions. We'll do our KS test and then get the p-value, save the p-value, get the means of each condition, and then do our log2 fold change, again, adding epsilon to the top and bottom of the fraction. And then save the log2 fold change. And then finally, after we do this for every gene, we'll cast our p-value and log2 fold change lists into NumPy arrays to just get them easier to work with. So yeah, this is really like the key part of the analysis, just taking what we've already done and just going through and doing it for every gene that we're going to be testing. So we can go ahead and run this now. Okay, now we're almost done. Almost done with this analysis. One more thing we need to do though, before we get to actually like checking the results basically. Yeah, one more thing we need to do. So if you guys remember what a p-value is from the statistics video, a p-value is the probability of seeing a result at least as extreme as the one you observed by coincidence if the null hypothesis were true. And sometimes people like to kind of put a threshold of 0.05 on the p-values as like a threshold for statistical significance, because a p-value of 0.05 or lower means that you'd only expect to see a result that extreme by coincidence only 5% of the time or less if the null hypothesis were true. So people kind of put that 0.05 threshold as like a threshold for statistical significance. Here's the problem though for using that as a threshold for what we just did. So if we did a test and we got a p-value less than 0.05, for the observed result for that test, basically, that would mean that it would be like statistically significant in the sense that we would only expect to see a result that extreme by coincidence 5% of the time if the null hypothesis were true. So yeah, that would make sense if we did one test. The thing is though, we just did about 11,000 tests, right? So yeah, how many genes did we just test? Yeah, we just tested 11,097 genes. So if we use a p-value threshold of 0.05, that would mean we would be getting quite a lot of coincidences of things falling below that threshold of 0.05 just based on the fact that we just did like 11,000 tests. Yeah, like the more tests you do, the more opportunity there is for there to be a coincidence. Does that make sense to you guys? Hopefully it does. If that's confusing, I realize maybe I'm like not the best at explaining it, but actually, I do have a link here to a comic that explains it, I think, even better maybe. So yeah, there's a really great comic that explains this problem with using like a fixed p-value threshold like this. So in the comic, there's some scientists investigating this hypothesis that jelly beans cause acne. And they basically decide to test if jelly beans cause acne. But they decide to test different colors of jelly beans. And they use this like basically for each test that they do, they use this p-value cutoff of 0.05 as like a significance cutoff. So they basically test a bunch of different colors of jelly beans, and for most of the colors, they get a non-significant p-value. So they get a p-value above their cutoff of 0.05. And they get that for most of the jelly bean colors that they test. But yeah, they do a bunch of these tests for all these different colors of jelly beans. And it happens to be that for green jelly beans, they happen to get a p-value that's actually below 0.05. And in that panel, someone actually says, whoa, because that's their cutoff for significance. So for green jelly beans, they do actually get a significant p-value. And significance here meaning you'd only see a result this extreme by coincidence 5% of the time. Like due to coincidence, if the null hypothesis were true, meaning if jelly beans actually did not cause acne. But yeah, they get that significant result, and they go on and keep testing and keep getting a bunch of other non-significant results. And then they publish their exciting result that they got a significant result that green jelly beans are associated with acne. And they put in the publication, I guess, only 5% chance of coincidence. Which actually, I don't actually like this wording here, because even it's the p-value of 0.05 doesn't mean there's a 5% chance that it was a coincidence. It means that if the null hypothesis were true, you would only see a result that extreme due to coincidence 5% of the time. So yeah, actually, even in this comic, I don't exactly like this wording, because yeah, do you guys understand that like subtle difference in wording? The p-value does not tell you the probability that something is a coincidence. It tells you the probability of observing a result as extreme as the one you got by coincidence if the null hypothesis were true. It's like subtle difference in wording there. But the point anyway is that in this case, this still is not very good evidence that green jelly beans are linked to acne, because they're using a p-value cutoff that basically says that for this cutoff you would only be expected to see results that extreme 5% of the time due to coincidence if the null hypothesis were true. But that's per test. And they just did like so many tests that the more tests you do, the more chance there is of seeing a coincidence. So if you're only likely to see a result that extreme 5% of the time or 1/20th of the time due to coincidence if the null hypothesis were true, but then if you do 20 tests, then it becomes like quite likely that you might get a result below that cutoff just due to coincidence if you do like 20 tests. You know what I mean? So basically the whole point of everything I'm saying here, the whole point of this comic and everything I'm saying here about these like p-values is that the more tests you do, you need to like adjust either your p-values or your threshold cutoff to account for the number of tests that you're doing. It's called a multiple test correction. And the type of multiple test correction we're going to do is basically the simplest one called a Bonferroni correction. So in a Bonferroni correction, you basically just multiply all of your p-values by the number of tests that you did. So in our case, we did a test for every gene. So we'll correct our p-values with this Bonferroni correction by just multiplying all the p-values by the number of tests that we did. And by the way, that's actually mathematically equivalent to taking your p-value threshold and dividing it by the number of tests that you did. So yeah, we could either use the same threshold and multiply our p-values by the number of tests, or we could keep the p-values the same and then divide our threshold by the number of tests that we did. So yeah, that's called a Bonferroni correction, basically the simplest type of multiple test correction that you can do. This is another simplification I'm making in this educational video because in real life bioinformatics analyses, they don't usually do a Bonferroni correction. They usually use this other type of multiple test correction called the Benjamini-Hochberg correction, which is a little bit more lenient. But yeah, for educational purposes, I decided to just go with the Bonferroni correction because it's like easier to understand intuitively, basically. So yeah, we'll do that Bonferroni correction to correct our p-values for all the multiple tests that we did. Okay, so now at this point, we've basically got what we need from this analysis, and we're ready to start actually checking some of these results and identifying some differentially expressed genes, which you guys might hear called DEGs for short, basically. But yeah, we're ready to now identify some of these differentially expressed genes based on their corrected p-value after doing that multiple test correction and the log2 fold change. So yeah, this block of code here basically again uses some of this NumPy slicing to basically make a Boolean array of true and false values for every gene, and it'll give us a true if the gene has a corrected p-value less than or equal to 0.05 and a absolute value of the log2 fold change greater than or equal to 2. Just as a note, this cutoff of greater than or equal to 2 on the log2 fold change probably is too strict and is stricter than would be used in a real life scenario. In real life, it might make more sense to use a cutoff of 1 maybe, or maybe even less than that. But yeah, remember a log2 fold change of 2 or negative 2, because it's the absolute value. That means that for this to be satisfied, the mean of one of the conditions had to be like 4 times the mean of the other, 4 times or more the mean of the other, basically, which is a pretty big difference. That's a pretty big difference in means. And yeah, it might make more sense to make this a 1, which still would mean that one of the means had to be twice the other mean. So even that might be too strict, to be honest. But yeah, the reason I'm using this for the video is actually for actually not a very good reason, but it's actually just because we're going to make a plot later on, and the plot will be easier to understand if we don't have like too many genes coming up. So yeah, it's actually not a very good scientific reason to be using this cutoff, but yeah, I'm kind of actually making it like more strict than it needs to be, just for the like plotting and like visualization we're going to do later on. Basically, I don't want too many genes in that plot, so I'm going to be a little bit stricter than I probably would be in like a real life analysis. But yeah, we can basically use this, like, again, NumPy like Boolean slicing to select the genes that have a corrected p-value less than 0.05 and an absolute log2 fold change greater than or equal to 2. And yeah, this block of code will do that. And then with this block of code, we can kind of like print out some little bit of a summary on what we found. So basically, with these cutoffs, we identified 75 differentially expressed genes. Again, there was a total of 11,097 genes tested, and the percentage of genes identified as differentially expressed was 0.71 something percent. And this is not 70%, this is 0.7%. So only a very small percentage of the genes tested came up as differentially expressed. Again, that's just because I made this threshold more strict than it probably would be in real life. Again, you guys can actually like play around yourself with like different threshold cutoffs here and see what you get. But yeah, I made it more strict than it probably would be in real life, just to really narrow down the list of genes. And we can tell, again, based on whether the log2 fold change was greater than 0 or less than 0, which condition had the higher mean expression, basically. So the term upregulated DEGs, or upregulated differentially expressed genes, means differentially expressed genes that had higher expression in the IPF condition. And then downregulated means differentially expressed genes that had lower expression in the IPF condition compared to the control. And yeah, we found 65 upregulated differentially expressed genes and 14 downregulated differentially expressed genes. Okay, so that's it for this video. That was our differential expression analysis. In the next video, we'll look further into these results and try to just explore these results a bit more and see if we can figure anything out about like the biology of these genes that came up as differentially expressed. So yeah, thanks for watching, and see you guys next time.